Welcome to the After On podcast. I'm your host, Rob Reed, and this is a series of conversations with thinkers, founders, and scientists. Take a little time and stretch out, because these talks are unhurried and meant to bring you to a top percentile understanding of something important. So, whether you're into startups or ideas, a techie or a lit major, take your time, engage your mind, and you'll be glad you did it. Especially this week, when we'll be talking to
Ed Boyden, who's one of biology's most influential young researchers. Indeed, Ed is one of the most influential researchers of any age in biology. Though he's not yet 40, the lab he founded at MIT is already the second largest at the entire institute. And Ed himself has already invented or co-invented a panoply of research tools that have spread to thousands of other labs and are collectively helping to change the face of life science research. These include optogenetics, a method for exploring and triggering neural circuits on a far more granular level than had ever been possible before. And also expansion microscopy, a delightfully inverted technique which, rather than magnifying the image of a microscopic object, expands the object itself to make it more visible. Ed will tell us about both of these breakthroughs and much more. But first, I'd like to welcome everybody to the third season of this podcast. During my 2-month hiatus from publishing new material, I've recorded and produced several fascinating interviews, some of which will be double episodes, which I'll present over 2 consecutive weeks because they just cover so much ground. I've also produced some fantastic material which I'll present to the generous people who sponsor this show on Patreon, including something particularly special today. And last but not least, this show has its very first sponsor. More about that in a second, but first, if you're new to the show, welcome. It features in-depth conversations with world-class thinkers, founders, and scientists about their domains of knowledge. In each episode, I try to bring you and myself a serious grounding in something complex and important that we should all probably understand a bit better. If you enjoy this episode, please browse the show's archives at after-on.com or in your favorite podcasting app. You'll find an archive of intriguing conversations with some of the top people in robotics, neuroscience, cryptocurrency, drones, augmented reality, synthetic biology, and many other fields. Now, back to my first sponsor. Long-term listeners will know that I've been holding off on launching advertising for the better part of a year. I only want to bring you high-quality and perhaps even innovative advertising that I personally believe in. That's a tall order for an independent show with a modest audience, so it took time to come together. But I think it's been worth the wait. For the next 4 months, this show will be sponsored by a venture capital firm called Obvious Ventures. You'll hear more about them in a couple of minutes, but first I want to tell you about the approach we're taking. Each sponsored episode will include a very compact mini interview with someone smart about something fascinating. That's, of course, the very thing this show strives to deliver, so these conversations will hopefully be every bit as interesting on a minute-per-minute basis as the show itself. For the first few sponsored episodes, I'll be interviewing investors from the firm about technologies that intrigue them. They'll usually mention a company that they've invested in passing, but the real point of these conversations is to present a cool, emerging field that most folks don't know about. We'll be discussing things like air taxis. Yeah, flying cars. Really. Which are coming a lot sooner than I, at least, ever imagined. We'll be exploring new manufacturing techniques, which build up valuable materials like diamonds on an atom-by-atom level, and a lot of other great topics. Later this spring, I'll start interviewing founders who've been backed by Obvious Ventures about the technologies their teams are developing. Then at the end of this sponsorship in June, the investors will be back to tell you about some emerging fields that they keenly want to invest in hopes that some of you will be inspired to submit business plans to them and perhaps go on to build a great startup with the aid of their capital advice and other resources. We'll now hear a 2-ish minute conversation with Obvious investor James Joaquin, which will give you an overview of the firm itself. Then after my long conversation with Ed Boyden, James will be back for another 2-ish minutes to tell you about a cool new approach to molecular manufacturing. After that, we'll have my normal closing remarks in which I'll tell you about the amazing guests we have lined up for March and April, and also about the very special content I've produced for the show's backers on Patreon this week. And here we go. So James, for those who don't know, describe what venture capitalists do.
Venture capital is about taking very, very high risk early stage bets on teams and ideas. And that's very, very different than traditional banking or lending. Our job is to really put our money at risk, which is why we try to put our brains and our time behind that money to help our founders.
Tell me about the creation of Obvious Ventures.
Well, Obvious Ventures started 5 years ago. I'm one of the co-founders. My other co-founders are Ev Williams, created Blogger and Twitter, and is now on his third act as the founder and CEO of Medium, and Vishal Vasish, who was the chief strategy officer of Patagonia.
And what's a typical size and stage for a company you invest in?
Well, our bread and butter is early stage investing. So we typically invest between $1 to $5 million as an initial investment. We like to see the founders in place, maybe a working prototype or some early set of technology that we can get conviction around and know that it's defensible in the market. We invest around these three pillars we call sustainable systems, healthy living, and people power. And within those, we have lots of investment themes that we pursue.
With a firm like yours, how long does your money typically remain invested in a startup after it first goes in?
The industry average right now is 7 years.
And how many investments will your firm make in a typical year?
We'll look at about 1,500 investment opportunities and we'll fund 10.
Your stated objective is to make world positive investments. Tell us about that.
Ev, Vishal, and I, we all found of the companies that we worked on, the one that created the biggest financial returns for us and our investors was also the company that had the biggest social or environmental impact. And we saw causality in that. And we said, when we look forward the next 20, 50 years, we think the biggest companies of our time will be purpose-driven businesses that are set out to reimagine huge industries in what we call a world positive way.
So thank you very kindly for hosting me today, Ed, here at the Synthetic Neurobiology Group, which I believe it's accurate to say you both created and now lead.
That's right, yeah.
I'd love to get a quick sense of what brought you here. What started you down the path of study and discovery, which resulted in the ultimate creation of this group?
When I was a kid, I went through a sort of philosophical phase. I was probably 8 or 9 and wondering about the meaning of life. And I went through a bit of a religious phase around that time as well. And in the end, decided that wouldn't it be great if we could understand the human condition through science, if you will. And so I got bitten by this bug to try to figure out if we could understand the nature of suffering and happiness and what should we do. And so I started college really young. I was 14 years old. And,
14? Really?
And I started working on projects at the border between philosophy and science. So I worked in a group led by a professor at the University of North Texas, Paul Braderman. And we started working together on the origins of life. Could you create life in a test tube? So again, very philosophical, but also very nuts and bolts science. And that was a really hard problem, which we did not solve.
Yeah, we probably would have heard about that if you had.
Maybe, yeah. And so 2 years later, I transferred to MIT, where I switched from chemistry into physics, and ended up working with Neil Gershenfeld on quantum computing, which again is sort of the border between the weirdness of philosophy and Schrödinger's cat and all that, and then something very practical, like a computer. That was also a very hard problem. And so 20 years ago, almost to the month, I decided, third time's the charm, why not the brain? And two things happened. One was all of my previous experiences in chemistry, engineering, and physics had prepared me to take a different point of view on the brain.
A different point of view from the typical neuroscientist?
Well, a technological point of view. If you wanted to solve the brain, you need better tools. And so where better to bring tools in than from physics and chemistry?
Now, in light of what you just said, I want to read out your lab's mission statement. It is revealing insights into the human condition and repairing brain disorders via novel tools for mapping and fixing brain computations. So, opening your mission statement with a call to reveal insights into the human condition seems to reflect philosophical interests that go clear back to your childhood.
Oh, of course, yeah. And my hope is eventually to return to such questions once we have enough insight. So, you suppose the first 20 years of what we do is all about tool invention. And now the second 20 years, which overlaps with the first a little bit, is about mapping and understanding circuits, ideally with enough precision to model them in a computer. And that third phase is indeed about computational neuroscience. Can we simulate thoughts and feelings? And then the fourth phase could be to enlighten and repair. And of course, along the way, we're always deploying insights if they can help people. We're not waiting until the entire research agenda is done. But a lot of these clinical applications are byproducts of this longer-term quest to understand who we are in a way.
And it's a very big team that you have here. I think I read somewhere it's the second largest research group at MIT. Is that correct?
Yeah, we have 50 full-time people graduate students, postdocs, research scientists, and so forth.
Wow.
And then we collaborate with people in probably about 100 research groups all over the world. So it's a very extended network that brings together every discipline of science, if you will.
Yeah, I think I saw you use the word omnidisciplinary somewhere once.
That's right. We're using ideas and collaborating with people from every part of the sciences and engineering, except I don't think we've used gravity waves yet, and we're unlikely to.
Well, at least we can detect them now. Now, you've sometimes spoken about a transition when a field essentially transforms from an art form into a highly productive, design-driven science. Could you maybe give a couple examples of times when that's happened?
If you look at chemistry after the periodic table versus beforehand, once you have a systematic framework for thinking about chemistry, you can make all sorts of polymers, crystals, semiconductors, you name it. Before that formalism, which involved several decades of synthesis, occurred, it was very difficult to create something new. Maybe you spend your whole career making one thing.
And I guess that one thing, like a single polymer or a single novel material, would be your masterpiece. That definitely would be more like art than science.
Or computer science is probably the most recent science to go through this transition. Now a high school kid can write an app and have it on 1 million devices in a few weeks because it's a design science. But in biology or medicine, you can have an idea for a cure. It can work in a cell culture. It can work in a small-scale human trial. And then you go and try to take it to the market, and it fails, sometimes for the most obscure of reasons.
So it hasn't obviously gotten to that point of being a very highly productive, data-driven science yet. What will we need to do in biology? What's the periodic table? I mean, in a certain sense, I guess you could say we've done it partly because we've cracked the DNA code and we understand how base pairs translate into genes, into proteins, and so forth. But what still remains to turn the biological sciences and the neurological sciences into a highly productive, data-driven science?
Well, just having the DNA sequences isn't enough, right? So if you think of the A, C's, G's, and T's of the genome as instructions for a cell, but then, of course, these genes encode for proteins and other machines that are actually making the cell do what it does, you really need to know what those are and how they work together. What's the difference between knowing that there's a lot of zeros and ones on my hard drive and then actually looking at a computer and typing an email and sending it? And Alan Turing and others actually made a formalism where you call, this, the halting problem, right? Where you cannot predict what a computer program is going to do in a fully algorithmic way. So you need to make what I call a ground truth map of the building blocks and how they interact.
Now, let's talk about that. You actually use the term getting to the ground truth more than most people, and I get the sense that you mean something very, very specific by it. What ground truths are we missing right now in biology to get it to this high productivity state?
Well, in chemistry, you have a list of the atoms or elements in the periodic table, and you have a list of the bonds that can exist between them. And then you have a list of the reactions that can take place. In physics, you have a list of elementary particles, electrons, protons, and neutrons, and the forces that they can undergo, electromagnetism, gravity, and a few others. So in biology, the question is, what are the building blocks and how they interact? The building blocks are the biomolecules, sugars and fats and proteins, and the list of those, which is probably in the tens to hundreds of thousands or millions of things. And then how do they interact? How do they talk to each other? And a lot of things are the building blocks that we're aware of. We don't know what they really do in many contexts. And then there are lots of building blocks that we're not even aware of.
So it's really a comprehensive map of that entire domain that still needs to unfold. What all those building blocks are, what the biomolecules are that we don't know about. And a lot of your focus has really been on the tools to get to those ground truths.
That's right. In our group, we've been working on tools to try to really map molecules throughout cells and cells throughout tissues and organs. We've been trying to work on ways to see with great detail the high-speed physiological dynamics of cells. And then thirdly, we've been working on ways to try to control those high-speed physiological dynamics. And so what I want to do in the years to come is not only to further those three legs of a tripod, if you will, of technology, but to combine all of them into an integrated pipeline for mapping and analyzing and repairing complex biological systems like brains.
With an ultimate goal, as you said, of solving the brain. Now, this is another term I've seen you use multiple times. What specifically do you mean when you say solve the brain?
Well, my dream would be to have the right kind of biological data, structural and dynamic, so that I could simulate in a computer, in a biophysically accurate yet human understandable way, how a brain circuit implements a decision or an emotion or some other complex function that characterizes, in the beginning, the processes of small brains, like animal brains, but eventually the human brain.
And I think it was in 2013, CNN quoted you as estimating this would take something in the neighborhood of 40 years. Do you now feel like we have 34 years to go?
In some regards, we're moving ahead of schedule because neurotechnology became cool. It didn't used to be cool. That's actually part of why I ended up at the MIT Media Lab, because a lot of the places that I applied to for jobs didn't think this was a ready, mature vision. But the MIT Media Lab and the MIT School of Architecture has had a long history of trying to hire misfits who don't quite fit into classical disciplines. But because the technologies we built worked, and then people wanted more of them, and then there were initiatives like the Obama Brain Initiative that poured a lot of money into getting people interested in technology and building technology. Over the last 6 or 7 years, especially, neurotechnology became cool, and lots of people joined the field.
So we're perhaps speeding ahead of schedule as a result of that.
Perhaps. Hard to know for sure, because this is real science, which means there's fundamental, unpredictable parts of it. But in our group, at least, we feel now that the tools are moving fast enough that it's time to start actually applying them in these combinations to solve problems.
One of the challenges that you face, perhaps uniquely in the brain, is that there's this incredible range of scales that it exists on and operates on, both temporal and physical. Could you characterize the sweep of that breadth and the challenges that it poses, both on the time scale and on the size scale?
Well, brain cells are enormous. Neurons are the largest cells in our body by far. In our brains, they're centimeters in spatial extent. And then if you include spinal cord neurons, they're like a meter long. Compare that to a blood cell or a tiny immune cell, which will be a fraction of a human hair diameter. Nevertheless, though, the proteins and the neurotransmitters and all the building blocks of brain cells are nanoscale. So how do you go from nano to macro? That's a unique problem in biology in the sense that you have to go through all these different spatial scales if you want to understand something as complicated as a decision or an emotion in terms of the elementary building blocks like transmitters, receptors, ion channels, and other molecular building blocks of brain cells.
And what's tricky is it's not just, I guess everything on a certain level exists on a nanoscale because things have small scales to them, but it's actually organized, like at the neural vesicles and so forth. It's actually organized on a nanoscale, despite having this immense extent.
That's right. You can take a molecule called a receptor. So these are molecules that listen to messages from other brain cells. And if it's at one point on a cell, it might drive the cell to be healthier, and it might drive the connections between cells to be stronger. Shift it by 100 nanometers or something, and now the signal means the opposite. Maybe now it becomes a signal of cell death, and when driven, it might actually weaken a connection. So nanoscale architecture matters.
And mapping that is particularly difficult because of the limitations of microscopy.
Well, light has a finite size, or wavelength. And what that means is you can't see stuff that's a lot smaller than the wavelength. Now, for visible light, blue, green, and red, and so forth, that wavelength is about a couple hundred nanometers, 400 to 800 nanometers, let's say. Now, a biomolecule, on the other hand, might be 5 nanometers in size, or even smaller. So much, much, much smaller than the size or the wavelength of light. In 2014, a couple of the inventors of so-called super-resolution techniques won the Nobel Prize in Chemistry for breaking that resolution limit, which is known as the diffraction limit. But these technologies can be quite slow and difficult to apply to three-dimensional structures like brain circuits. And so, for the most part, nobody had really used these techniques to map an extended object like a three-dimensional brain circuit. It could take a long time to actually map such a thing, or a daunting expense, which means it hasn't been realized yet.
How is super-resolution microscopy different from electron microscopy?
So in electron microscopy, you stain a specimen with an electron-dense reagent, and then you scan an electron beam, and that gives you the contrast. So it gives you basically one color, the contrast of the electron-dense material. With light microscopy, you could have many colors, blue, green, red, and so you could potentially see many, many kinds of information in a specimen at the same time. Playing clever physical and chemical tricks on either the dyes, the stains that you use in light microscopy, or on the physics of the light that you deliver. But because those are physics tricks, they're rather elaborate and hard to make more parallel. Wouldn't it be great if you could make microscopy faster just by, let's say, doing more engineering rather than having to pioneer more physics?
Now, the temporal scale is in some ways even more yawning.
Yeah, so the electrical pulses that brain cells make and the chemical exchanges that brain cells talk to each other with, those are milliseconds or less in duration, and they're timed with millisecond precision. So the issue there, of course, is that you need to look at these very high-speed biological signals, much, much faster than the development of the kidney or the movement of an immune cell or other kinds of biological process. And yet, if you care about learning or Alzheimer's disease or changes in emotional state, those can take place over months, years, or even decades.
It's astounding to me to think about things that are happening in a millisecond accruing over a time span like 80 years for something like Alzheimer's. I mean, that does introduce an incredible complexity of scale. I did some goofy back-of-the-envelope math last night. If the most fundamental granular events happen at the millisecond scale, if Alzheimer's develops, let's say, over 80 years, that's something like 165 billion of those events leading to that one outcome. And the goofy analogy I came up with, because the Super Bowl was last night, and to absolutely nobody's astonishment, your local team, the Patriots, won for, I think, the 600th consecutive time.
Mm-hmm.
If the fundamental granular event in a football game is a play, there were about 150 plays in last night's game.
Okay.
On a similar scale, that would have been a 315,000-year game.
Wow.
There'd been 165 billion of those things.
Mm-hmm.
Obviously, it's a bit of a goofy analogy, but it just puts into perspective the yawning number of fundamental units of action and activity that go into some of these processes.
Oh, yeah. And the spatial scale is pretty daunting as well. If you took one human brain and scanned it with the resolution of an individual transmitter or receptor, and then you took all that data and bought those little USB hard drives off the internet and loaded all the data from those images in there and stacked those hard drives up in a tower, that tower would, for one human brain, would reach from the surface of the Earth well into outer space.
Really?
One brain.
One brain. So let's talk about expansion microscopy.
Sure.
Attacking the spatial challenge. Could we start with just a quick overview definition of what it is?
So for 300 years, people in biology and medicine have been using lenses to zoom in on cells. And that's been great. It's yielded the germ theory and discovery of the brain cell in the first place and untold advances in biomedicine. But as we talked about earlier, it doesn't get you down to the nanoscale. And the super resolution methods use clever hardware and physical tricks, but as a result, they can mostly be applied to fairly thin, two-dimensional small specimens, as opposed to large, extended 3D specimens. So we started thinking, what if we did the opposite? We could take a piece of brain tissue, preserved, of course, not alive, and if you infused it with the right kind of swellable chemical, like the stuff you find in baby diapers, maybe you could add water, swell the polymer, and blow the brain up until it was 100 times, 1,000 times, 10,000 times bigger in volume.
So instead of magnifying it optically, magnifying it physically.
Correct. And then you could use cheap optics, like the kind that laboratories already have, or you could design through the principles of engineering very fast optics in order to scan essentially as fast as you wanted.
And the inspiration for this, some of the papers were quite old, from the early 80s, that initially gave you some of your original ideas for this?
Yeah, well, there was an MIT professor named Toyoichi Tanaka, who was one of the pioneers of smart gels, responsive polymers. These are polymers like the active ingredient in baby diapers, will swell enormously when they absorb water or other solvents. And we started reading papers from him, and we decided, what if you could embed polymer like that inside the tissue in an even, dense way and chemically treat the tissue to soften it? Could you then blow it up? And two grad students, Fei Chen and Paul Tilburg, started doing this. And within a few months, we got something like that working pretty quickly. And it helped because there actually is a long history of people doing hydrogel polymer embedding of specimens. So people like Peter Hausen and Christine Dreyer in the early 1980s at the Max Planck Institute were taking embryos and tissues and all sorts of specimens that they had. They were studying amphibians and doing hydrogel polymer embedding and then using that to facilitate microscopy. So you could take our technology that we call expansion microscopy and say, hey, it's kind of combining this idea of hydrogel tissue embedding, which goes back to 1981, and the idea of hyperswellable polymer physics, which goes back to around the same time.
But we live in a different world today, and there's technologies that we have that perhaps that are enabling that they wouldn't had access to back then. Or could this have been done, do you think, in 1981?
I think it could have been done, potentially. The tricky part of neuroengineering is the neuro part, understanding the need that you're trying to solve. To be honest, when we first cooked up the idea in the group, we kind of didn't do anything about it. So I was brainstorming with one of my first postdocs in 2007, and we were trying to figure out how would you decrowd the molecules in a compact environment to make more room around them, move the molecules apart in an even way to make room for tags that you could bring in.
So kind of like putting dots on a balloon and blowing up the balloon and getting space between the fundamental units. You'd still have dots on the balloon, which would still be the biomolecules, and their configuration is preserved. Their relationship in 3D space is preserved. The biomolecules would not be spread out, but the space between them would be expanded.
That's the idea, yeah. But that was in 2007, and 1 year before, some of these super resolution techniques had come out. So
yeah, you're right in the midst of that.
We didn't really do anything because there are a lot of people in this field. But 5 years later, when Fei and Paul joined the group, they actually were trying to do super resolution and electron microscopy, and we realized that, hey, if you really wanted to map a brain, you might need something new.
And then also super resolution and electron microscopy, I'm sure, is not in the budget of every single lab everywhere. There's probably an element of access. that when you blow things up to the point where they can be viewed with a light microscope, suddenly that kind of work can be done in a lot more places?
Oh, yeah. I mean, great biotechnology will be easy to use, right? I mean, if you look at the history of biology, the great technologies are always really, really simple. You know, green fluorescent protein, which must have been used in 1 million studies. You put the gene in and it works. CRISPR, right? Everybody's heard of CRISPR nowadays. You put the gene in and it works. I think you see the pattern here.
Yeah.
So great biotechnologies, for the most part, are simple and reliable and inexpensive. And I think there's something to it, more than just ease of use. Another reason is that biology is complicated. So for a biotechnology to work well, it better be simple or it won't be robust enough to work in a real system. So I sometimes call this the principle of applied laziness. A great biotechnology should be simple and easy because otherwise it might struggle to actually gain widespread adoption.
Now, the idea of taking something tiny and expanding it does sound very sci-fi. So could we go briefly through how does it work, sort of step 1, 2, 3?
So you take the specimen and you add anchors that bind to DNA, RNA, proteins, other biomolecules.
So every molecule has an anchor on it.
That's the hope. Yeah, we haven't checked every molecule, but we're trying to get a dose which will cause the majority of the molecules to have an anchor. And then we add what are called monomers. Those are the building blocks of the polymers, and we wait till they evenly diffuse throughout the specimen. And then we trigger the monomers to form polymers. Basically, they line up into long chains and form like a dense 3D spider web throughout the specimen.
And there's like a catalyst you add that starts the monomers turning into polymers, or is that something that they just do naturally?
Well, there's a chemical called an initiator, and you can also change the temperature to make the chemical reaction run faster.
So in a sense, you're infusing this sample that you want to expand with a whole bunch of tiny little strings that once they're activated will start linking together and winding around and becoming more like a web or a network that can pull things apart. Is that a reasonable analogy?
Kind of, yeah. In reality, it's more like we're flooding it with little ping pong balls, and then they self-assemble into strings of ping pong balls. So then it looks like a spider web, a 3D spider web, where these ping pong balls are the actual wires of the web.
And now that the biomolecules, what you want to do is pull them apart so that you can see them in their individual glory as opposed to as this indecipherable wad. Is it like a just add water thing, literally? When I think about a baby diaper, you just add some, liquid, and it expands. Is it as simple as that?
Yes, except we do one step before then, which is we soften the tissue so that when the baby diaper-like polymer expands to push the biomolecules apart, the natural forces of the tissue won't resist. So we can use detergents, enzymes, other chemicals to soften up.
Loosen it up.
The specimen. Yeah, so it's like meat tenderizer. You add some enzymes, softens it up. You can use heat, kind of softens things up. And then when you add water, it'll expand.
And what does it do that Electron and other pricey microscopy formats cannot? I know it makes the molecules a lot more identifiable.
Well, there's two payoffs here. So one is resolution. If two biomolecules are too close, closer than the resolution of your microscope, you can't tell them apart. You can't tell if there's one thing or two things or a smiley face or a pyramid because they're all jam-packed together. So by separating them, you can see them better. So you might have a bunch of things that are too close to resolve. You expand them and now you can see the shape. Oh, it does look like a smiley face or a pyramid or whatever. The second is decrowding. So if you have a bunch of molecules jam-packed together, you might not be able to access them for labeling. When you see a biomolecule, you're not looking, for the most part, at the actual biomolecule. You stick on a little tag, and the tag has a glowing dye, and that lets you identify and locate the biomolecule.
A certain tag, a glowing dye, is going to be drawn to a particular molecule.
Exactly, yeah. And so you can buy lots of tags that bind to different biomolecules, but if there's no room for them to get into a complex of jam-packed together stuff, then you won't label it.
What are some of the practical and research applications that are currently underway using expansion microscopy? And when did you first put it out there in the public realm for people to adopt it in their own labs?
So we announced the discovery that you could evenly expand a biological specimen in January 2015. And then a year later, we had a couple papers showing how to do it more practically. And then a year after that, we both showed how to expand things to make them really big. So you can get down to almost single molecule resolution. Not quite. We're getting there still. And also that year, we showed that you could apply it to human tissues. So one big question is, can you detect diseases early? Many diseases, if you detect them earlier, you can help save lives before it's too advanced. So we showed that we could actually do things like expand cancer biopsies from human patients and detect diseases at a point in time, which confounds many pathologists and just looking at the specimen with a conventional microscope.
So that's a very powerful practical application. And that's come up very quickly. I mean, it sounds like this has been out in the wild for maybe 2 to 3 years, depending on how you measure it. Is that currently very experimental, or are there tumor committees someplace that are already using expansion microscopy, maybe with AI or something, to find strange deformities within a tissue that might be indicative of cancer?
Well, at the research level, many people are doing just that. We've trained probably between 300 and 400 research groups already on the expansion protocols, and many, many more learned it on their own by looking at our website, where we post all of our step-by-step protocols. And I co-founded a small company now that's trying to figure out if we can do early disease detection with expansion microscopy.
And diseases beyond cancer as well, like epilepsy or other things?
Well, other people have already started to apply expansion microscopy to human epileptic brain tissues.
Oh, really?
The expansion microscopy is kind of out there now. I mean, the last couple of months alone, Michael Rosbash, who won the Nobel Prize for his work in circadian rhythms, has been using expansion microscopy to map out how some of the neurons involved with circadian rhythms couple to and control the neurons that actually control sleeping and waking.
So this is the intersections of the neuron's neural circuit can be understood on a more granular level as a result of this.
That's right. Brain cells are elaborately shaped, and they're connected by connections called synapses. The brain cells are called neurons. And within the neurons, you have electrical signals, and between the neurons, you have chemical signals. So the first order, what you really want to do is to map out how that thing is organized.
And that is something that is generally on too compressed of a level to be done accurately without something like expansion microscopy.
Yeah, it's really, really difficult. You can do electron microscopy where you image one two-dimensional section at a time, but it's very hard to get molecular information because there's only, again, one color primarily in electron microscopy. And then with super resolution light microscopy, you can get pretty good resolution, but it's primarily for small two-dimensional things. It struggles really to image large three-dimensional things.
It sounds like when humanity truly gets down to the serious business of mapping the human brain, this could turn out to be one of the 2 to 3 critical tools in our toolkit.
The tools are going to be very useful for a lot of brain mapping enterprises all over the world. I mean, we've taught people from many countries how to use it. It's so simple. We've taught undergraduates and people from all over the United States and beyond how to do it, and even non-neuroscientists. So we're collaborating with people on tuberculosis, on HIV, on all sorts of important biomedical problems.
Is it possible to expand something as large as a virus or a bacterium in total?
So people have already done that. A paper was published by a group at Harvard where they looked at the bacterium E. coli with expansion microscopy. And then a couple papers, I believe, have either been submitted or posted to the bioRxiv preprint server looking at viruses and their infections into cells.
And so a full virus could be expanded using this technique and understood on a more granular level than would otherwise be possible.
Potentially, yeah. I'm trying to think of if anybody's expanded an individual virus and seen an individual virus. People have expanded cells expressing viruses, but definitely it's the kind of thing that'd be very exciting to pursue.
Now, one of the things you spoke about in one interview was the potential, after something has been expanded, to do analysis on something as simple as a smartphone. That feels very revolutionary in clinical settings and other places where they're simply never going to have a gazillion-dollar microscope. Where does that stand in terms of development?
So that was the study actually from Harvard where they expanded a bacterium, Shrike Zhang and Ali Karamosseni, and they then used a modified webcam to take a picture of that expanded bacterium.
Another application I've read of is mapping chromosomes in 3D space. I mean, we typically, or at least I typically, think of a chromosome very simplistically and very inaccurately as being kind of like a line. But it isn't. It's a contorted shape, which I imagine is pretty hard to get a handle on, and that can impact how things get expressed, right?
Yeah. Well, the genome is a three-dimensional thing, and it coils upon itself in very complex ways, and distant parts of the genome can be close by and actually affect each other. Distant in the linear sequence,
right,
but close by in 3D space, to be precise. So seeing the shape of the genome can be quite powerful. And in fact, several groups have already started to use expansion microscopy to look at processes such as what happens during cell division as a chromosome separate. So it's already happening.
Now, there's something that you mentioned in one interview, in situ sequencing with George Church. Mm-hmm. Could you explain what that is and what its promise is?
Well, to sequence a piece of DNA, you copy it. And as you copy it, you're adding fluorescent or glowing DNA bases. So as you copy a piece of DNA, it's going to basically blink out the genetic code over many cycles. Now, in a sequencer that you might buy from a company like Illumina, that sequencing is done by a microscope and a robot that's on some kind of two-dimensional surface. But George's group showed in cultured cells that you could do this right inside a cell. In other words, you could sequence a piece of nucleic acid right inside a cell by basically copying it and letting it blink out its identity over time. So we thought, what if we can adapt this to brain circuitry and other kinds of complex tissues by expanding the brain tissue first and then running the in situ sequencing right there inside the expanded specimen? Then you could actually read out the recipe of a cell, if you will.
When you say the recipe, you mean which proteins are being expressed and which ones are not?
Which genes are on and which genes are off. So when the DNA of a cell is turned on, you get a molecule called RNA. And RNA is just about as easy to read out as DNA. So let's read out which RNA molecules are on, and we'll tell you which genes are on and which genes are off.
And here's kind of a dumb, big picture question, but of the 30,000-ish genes that the human genome has, in a neuron, how many are being expressed typically at any one time?
Well, there's lots of groups that are doing RNA sequencing of individual cells at the Broad Institute and at the Allen Institute for Brain Science. And I forget the exact distribution of genes. Obviously, some genes are very highly expressed, some are very weakly expressed. So there's a bit of a threshold. Where do you call that as a gene that's important to the neuron? And we know that there are very, very low copy number genes that might be very, very important. So when you ask the question of how many genes are on, quote unquote, in a cell, it's actually a very subtle question.
I imagine, the genes are going to tell my body, every cell in my body, how to do everything that needs to do. It feels like just intuitively in one given cell, a relatively small percentage of those genes are going to be in play. Some of them are just going to be doing things in my liver or my kidney or something like that.
Again, it kind of depends on how you define the cutoff. Because of biological noise, a gene that's quote unquote off might actually be expressed at some low level.
At a very low level.
Stochasticity. And so I guess the question then is, how do you define when a gene is on? And so that's actually a big biological question right now, where you might have a low copy number gene expression profile, but that gene, every copy might count.
But then in situ, you can figure out exactly what's happening within the cell, whereas before that happened, was it really a matter of guesswork?
Well, no, you can still sequence single cells from the brain, but you'd have to take those cells out. So now you no longer know where in the circuit they are. I think if we could figure out not only how the brain is wired up, but what molecules are in those wires, then you could have a much more complete picture of how the brain works.
So moving on to the temporal challenge, that gets to optogenetics, which is something that you co-invented prior to expansion microscopy.
Yeah.
Could you describe that in big picture terms, and then we can get a little deeper into why it matters so much?
When I started grad school at Stanford in neuroscience, after having trained in chemistry and physics and so forth, I was talking to one of my fellow students, Carl Deisroth. We were both working in the same group, and we started brainstorming. How would you actually control a brain cell? If you
Control it.
What we wanted to do was to control specifically the electrical activity of a brain cell. And at the time, lots of people were exploring how to use electrical pulses or magnetic pulses delivered by implants or delivered from outside the brain to sculpt brain activity. So deep brain stimulation has been used in perhaps 100,000 patients already, where you plant a wire in the brain and deliver electricity deep into the brain.
And that's often used clinically for things like Parkinson's, right?
And other movement disorders, exactly. But the problem is the electricity goes in all directions. So if you want to study how the brain works, it's not a precision tool. It won't allow you to activate exactly a pattern of brain activity or all the cells of one kind. So Carl and I just started brainstorming and just going through all the laws of physics. Could you use mechanical force? Could you use magnetic beads? And we decided that if you could use light, that would be the best.
This is when gravity waves was not on the table.
Yeah, plus the fact that the gravity waves are way, way off the energy and all the other scales.
Yeah, a little hard to direct those things.
Yeah, you need planet-size or city-size or country-size detectors even to see them. So we thought light would be great because it's as fast as anything ever gets, and you could aim it if you want. So then the next question became, do we build molecules that convert light into electricity, or do you find them? And I'd read this paper from a group in Japan that talked about a class of molecules called Microbial Opsins. And so these are molecules that are kind of at a glance like the molecules in our own eyes that sense light. But rather than causing a chemical cascade, which is what our eyes do, these molecules convert light into an electrical flux, an ionic flux directly. And so at the time, these molecules were known to be from so-called halophilic archaea, single-celled microbes that live in really salty water. But this paper I read suggested that there actually were some of these molecules that worked in low salt, like the kind you'd find in your brain. And so that's what triggered us to start collecting these molecules from colleagues. And in 2004, Carl put one of these genes into some neurons, and I did the light shining and recording. And lo and behold, to our surprise, it really worked on the first try.
Wow.
We were able to activate electrical pulses in brain cells with pulses of light. And so that was very serendipitous. The molecule could have been toxic. The molecule might not have worked. Maybe it would have been too slow. Maybe it would require some chemicals to be added, which makes it much harder to use. That was one of my biggest worries at the time, because people have been studying these molecules in oocytes, frog eggs, for many years. And the frog eggs, you actually have to add some chemicals to make it work. So maybe if you had to add a chemical to make it work in a brain cell, maybe it'd be too difficult to actually use in an awake, behaving brain. But serendipitously, it all worked, and we didn't need to add any chemicals.
So when you get one of these genes into a neuron, suddenly something that would normally be on the surface of this single-cell bacteria appears on the surface of the neuron.
The molecule that we actually ended up trying out first in neurons was from the eye spots of green algae. So it's actually from an intracellular organelle, not the boundary of the algae.
Okay.
But somehow it manages to get to the surface of the neuron, and it's fast and safe and doesn't need any chemicals to be added.
So now that thing that in the algae would detect the presence or absence of light is obviously doing the same thing on the surface of the neuron. It's detecting the presence or absence of light.
Mm-hmm.
Is it something kind of like a pore?
Mm-hmm.
And what connects that pore to the inner machinery of the neuron that then causes the neuron to do something that you want it to do when you shine a light on it? Did that pore somehow get hooked into the physiology of the neuron in such a way that it could control the neuron?
Even simpler. So when a neuron is active, like in your auditory system when I say these words, or in my motor cortex when I say these words, it fires an electrical pulse. And what that means is positive charges will go into the brain cell. And that's called an action potential or a spike.
And these are the things that happen on the millisecond time scale that we were talking about earlier. Yeah.
Yeah. So the molecule that we put into the neurons, when you shine blue light on it, that was the original molecule. Now we have ones that respond to red light as well. It opens a little pore and positive charge flows in through the pore. And so that is the action potential or the spike. Or you can trigger an action potential or spike to occur by engaging the neuron's intrinsic electrical machinery to kick off.
So it basically puts it into a state where that spike is going to happen when and only when you want it to.
Yeah, in a little bit more biophysical detail, why does a neuron spike normally? Well, there are synapses, chemical connections onto the neuron, which release chemicals, and those chemicals will open pores and let positive charge in. If you let enough in, it crosses what's called a spike threshold, and a positive feedback loop gets kicked off, and you get this big electrical spike, where lots of positive charge goes in. So when we shine light on a cell with our light-activated pore, we are triggering that initial step that kicks off the positive feedback loop, and then we get the spike.
And then it happens. Now, when you get to, let's say, a small mammal like a mouse, do you treat them in such a way that 100% of the neurons in that critter, or something close to 100%, now have that pore on them? Or are you able to target it to particular neuron cell types? Or do you target it to very narrow geographies within the brain? How much granularity are you able to exercise when you decide to make the neurons of a particular creature's brain sensitive to light?
Depends on your scientific question. Maybe you want to activate a specific set of cells that's known to be atrophied in a disease and see if you can repair the disease. Or maybe you want to activate some neurons that you saw that were recently active and see what they do. Or maybe you want to activate a certain kind of pathway between two parts of the brain and see how they talk to each other. So it really depends on what you want to do.
So you are at a position where you can say, we want to put these activation pores only on, parasol cells or only on basket cells, to say we're really just going to activate this one cell type because that's the one that we want to spike when we apply light?
More or less, yeah. We don't have a full list of the cell types of the brain yet, so maybe there are cell types that are hard to target. But a lot of cells are defined by one or more genes that are on very highly. And if you use tricks from gene therapy that say, hey, any cell that has gene X on, turn my gene on too, then you can activate cells of that class.
Then how is the light imported into this brain?
Well, that's the easy part in a way, right? LEDs, lasers, there are all sorts of ways to beam light at things. And you can couple them to optical fibers.
So this will be like an implant that the creature, the mouse, has.
You can put an optical fiber into the brain.
And then what are some of the vivid and fascinating research applications and practical applications that have happened?
Well, there are two kinds of optogenetic molecules. One class lets you turn neurons on, and the other class lets you turn neurons off. So by activating a set of neurons, you can figure out what behaviors or pathological states or health-promoting states in an animal model, not a human, of course, that you could actually initiate or sustain. And by turning neurons off, you can figure out what behaviors or pathologies or healthy states they're needed for. So people are activating sets of cells and trying to trigger the recall of a memory or the initiation of a behavior, even a complex one like aggression or violence in mice. By turning neurons off, you could try to delete a memory or shut down an epileptic seizure or otherwise silence the activity of neurons to figure out what they're needed for.
Could we talk about the science behind what is now cognitive therapeutics? Because I think that's a particularly fascinating application.
So my colleague Li Wei Sai here at MIT used our optogenetic technology to make a pretty stunning discovery. If you drive the brain at a specific frequency, 40 times a second, which is sort of a magical number in neuroscience, then the brain's immune system turns on and other changes occur. And in mouse models of Alzheimer, so these are mice that are genetically engineered
to get amyloid plaques and other molecular hallmarks of Alzheimer's, and they have cognitive issues as well. Anyway, in these mice, they get better.
When you say drive the brain at 40 times a second, I assume that means you're flashing the light at 40 times per second. And are you bathing the brain in it, or is it a particular narrow region of the brain that is relevant in the case of Alzheimer's and in these mouse models?
In this case, it indeed was targeted to specific circuits, yeah.
So a narrow geography within the brain?
As a matter of practicality, yeah.
And so would it be just a handful of neurons that were being stimulated, millions of neurons? How many would be getting activated with that 40 per second pulse?
Oh, let's see. I'd have to think, but probably in the thousands to millions.
Probably in the thousands to millions. So now that pulsating happens, and what positive impact does the immune system bring in? It starts mopping up actual amyloid plaques, or?
Correct.
It does.
Yeah. So microglial cells, which are some of the immune cells of the brain, they go into a so-called phagocytic state. So they start gobbling up debris.
They're stimulated by that rhythm to start gobbling.
Through unknown mechanisms, yes.
Wow.
Yeah.
Now, you said that it's sort of a magic number in the brain. What else does 40 pulses per second do?
40 hertz oscillations are well known to go up during certain kinds of attention, for example. And in this study that Li Wei's group led, it also became clear that in these Alzheimer's model mice, the 40 hertz or 40 cycle per second oscillations are down. They're actually atrophied.
Their ability to get themselves to that level of oscillation when they focus, when they concentrate, has been atrophied.
Or at least the level of 40 hertz oscillations observed during awake behaving rodent activity was less.
How much treatment would they be getting? Would they be getting this 40 times per second activation for hours per day, just momentarily?
There was like half an hour to an hour per day for up to a week or so.
And then just after a week or so of that, they were able to see these amyloid plaques literally just break up and get mopped up.
They decreased quite a bit, yeah.
Were they able to figure out if the actual clinical signs of Alzheimer's were disappearing in the mice?
So the first paper is focused on the molecular hallmarks, including phosphorylated tau and other molecular hallmarks of Alzheimer's disease. But there are ongoing studies led by her group that are continuing to probe things like behavior and so forth.
To see if their cognitive function gets better. Now let's get to part two, because that is fascinating. Now on the non-invasive side.
Right. And this is so much easier to do non-invasive that
Yeah.
most of the studies that we're doing nowadays in this collaboration led by Li Wei are using the non-invasive method.
Mm-hmm.
So, Emery Brown is an anesthesiologist and expert in brain rhythms and made some suggestions to the teams, which led them to contemplate trying to deliver this oscillatory stimuli through the eyes. No optogenetics, no molecules from microbes, no optical fibers implanted in the brain, but just effectively a movie or a blinking light. And lo and behold, if you blink a light at the right frequency and have an animal watch it, again, no surgery, no optogenetics, no implants, that was also enough to get this 40 hertz oscillation going in the brain. And that was also enough to get the molecular hallmarks of Alzheimer's disease to go down in these transgenic mice.
That's amazing. What's the status of that research?
Well, the next stage is to understand the mechanism better of how it works and also to do human trials. So we've co-founded, Li Wei and I, a company called Therapeutics, which has begun human trials.
Really? Obviously with a non-invasive.
With, flashy lights and so forth. And it's early stages, but lots of stuff does not translate from mice to humans. So who knows what the outcome will be in the end. But if it's as simple as a flashing light, we'd be idiots not to try.
How do you trigger aggression with optogenetics in mice? You'd mentioned that study, and I'd read a little bit about that as well. They'll even attack a rubber glove, if I'm not mistaken.
Well, this is work led by Dayu Lin and David Anderson, then at Caltech. Dayu now runs her own group. And they found a cluster of cells deep in the brain that were active when male mice would fight. And so they made those cells light activated using our optogenetic technique, implanted an optical fiber aimed at those cells that was also connected to a light source. And they showed that by shining light on those cells and driving them, you could actually trigger a mouse to be aggressive and even attack things next to it, even if it was not an animal, even if it was just a rubber glove. So optogenetics lets you prove sufficiency of a given set of cells to initiate, at least, a behavior.
And that's pretty intriguing. I mean, getting back to your philosophical interests that we talked about at the beginning of getting to the bottom of human nature. I mean, aggression is something that leads to so many consequences. And not that any person has ever had aggression triggered within them via optogenetics. But when you start getting into these circuits and how they're stimulated, that might have all kinds of interesting philosophical ramifications for criminal activity and how we deal with it.
Questions that groups are trying to answer nowadays include, when you activate these cells or other cells in the brain, what's actually happening? Are you triggering a movement, and that is manifest as aggression? are you triggering a sensation? And now the animal's afraid and is aggressive in response to that. Or are there even more complex scenarios? Like, are you triggering some kind of reward in being aggressive? That gets into philosophical questions about what is justifiable in terms of aggression versus not justifiable. So those are very, very interesting kinds of studies that start to probe at the core of what all this means at a neural circuit level.
Now, way back in your TED Talk in 2011, you had speculated about the possibility that we might one day have optical prosthetics. Is that something that you still see perhaps being in the pipeline?
These are molecules from bacteria and algae and other critters that are not normally found in the body. So the big question is, will the body detect these as foreign and launch an immune attack against them? And so I view these primarily as a tool for science in the sense that we can use it to study the brain. And then, as with the study led by Liwei, can we then develop non-invasive or other deployable methods to get these things out into helping people? But there are a couple of groups that are planning human trials or even beginning human trials. But again, biology, as we discussed earlier, is not a full design science where you can predict the outcome of an experiment by thinking about it. And so if those experiments go well, maybe lots of people jump on it. But if they don't, then it'll stay as a powerful tool for science.
Now, to go back to your term of solving the brain, if the brain were solved, is that the sort of thing that would put us into a position to get very, very close to solving a problem? Maybe not all the way there, but very close to it by thinking about it.
If we had a model of the brain that had predictive value.
Which is part of your definition of solving the brain, if I'm not mistaken.
That's right. We want to have a biophysically accurate model that's ideally human understandable. But suppose that you can initiate the model and then run it for a little while and see what it would predict you do next, right? That could be really, really useful and also provide a way to validate the model.
It's making it clear to me what you mean by the transition of a science like into chemistry. Now we can figure out what's going to happen in a chemical reaction with pretty good accuracy before we actually do it. And therefore, industrial processes and chemical processes and scientific processes can be designed with pretty good rigor and pretty good safety.
Certainly a lot better than 100 years ago. I mean, chemistry is still more of an art than physics, let's say, or mechanical engineering. If you design an engine or a lever or a gear in a computer, you kind of know how it's going to work on the outside.
Yeah.
Chemistry, it's still more degrees of freedom than a mechanical engineering device, but certainly far fewer degrees of freedom than
we have here.
System.
It's interesting to think about. I think it's something like 75,000 people have Parkinson's implants now. Those are all electrical, right?
Those are deep brain stimulators.
Those are deep brain and electricity, and electricity has that property that you'd said previously. It goes in all directions. Obviously, they're implanting it in the point where they want to stimulate, but their level of granular control is going to be limited. So at least theoretically, optical prosthetics could do potentially a better job of that.
If it works. Again, you don't know if it works until it's worked, right? That's the problem with biology and medicine. Again, though, I think that non-invasive has a lot to say for it. If you look at gene therapies that are on the market right now, they're very expensive. And a couple of years ago, Nir Grossman, a postdoc in my group, and we published a paper where we asked, could you actually focus the effects of electric fields deep into the brain without surgery? And so we showed through a strategy that we call temporal interference that you could actually focus the effects of electricity to deeper structures without affecting the overlying structures. So what if you could have a non-invasive method that doesn't require surgery, that doesn't require implants or brain displacement or brain damage, which also could be more accessible and inexpensive? If there's a way to take your ultra-precise invention, like optogenetics, and then give it to thousands of groups to make discoveries with it, and then deploy inexpensive, non-invasive methods, that might be the fastest pipeline to get accessible therapies of high efficacy out to the largest number of people.
So let's talk about this Nir Grossman project, because it is fascinating. This is the one which you've compared to an AM radio, am I right? It's for like a low-pass filter.
Well, we've known that neurons are nonlinear low-pass filters for many years.
And for people who don't know what that means.
So electrical signals can have different frequencies. They can be low frequencies, like the same frequencies as the sounds of our voice or of music. And then there's really high frequencies, which is how you beam television and radio across cities, right? And then there's lots of intermediate frequencies. So brain cells fire around, let's say, 10 to 100 hertz, 10 to 100 pulses per second or slower. And the pulses are about a millisecond or so in duration. So fairly low frequencies.
Mm-hmm.
So that is evidence of their low-pass nature. They're not going to fire at arbitrarily fast speeds. And neurons are also known to have nonlinear properties. What does that mean? Well, in a mathematical sense, linearity means if you add two signals, you're going to get some linear combination of those things. But a nonlinear system will multiply them. Or in frequency domain, you might subtract two signals from each other. So we've known that neurons have this property for a long time. And so now the question is, are the properties good enough or appropriate enough that you could use them to do non-invasive deep brain stimulation. And what we found is a discovery that they are.
How do you trigger a specific geography using this technique deep inside the brain without having to do optogenetics or anything else that's invasive?
We're still working out the mechanism, so this is a little bit of an analogy than a proven fact, but let's think how an AM radio actually works.
Yep.
You beam the high-frequency waves across the city, and the AM radio is a nonlinear low-pass filter. So what that means is you're going to subtract off the high frequency and recover the voice or the music that was broadcast from the station. So let's now talk about the brain. We apply multiple high frequencies to the brain, and the brain ignores them the same way that we ignore the radio waves going across the city. But where these high frequencies overlap, we'll take the difference. The same way that the AM radio subtracts off the high frequency and gets the voice, the brain cells will subtract the two high frequencies and recover the low frequency, which, if it's low enough, could drive the brain cells.
And you can target that geographically or you cannot?
We can target it somewhat. Again, since
yeah.
we're not in the far field, in the spatial domain, these are fairly low frequencies from an EM, electromagnetism point of view. They're fairly large chunks of the brain currently. But our goal right now is to understand the mechanism. And if we can understand the mechanism, we could probably build much more focal versions.
So in theory, if you were able to build a version that was very focal, you might be able to stimulate that tiny quadrant deep inside a mouse brain, let's say, that triggers aggression, gets it attacking the rubber glove. Currently, we're doing that with optogenetics, which is invasive. And if you could do this with good focalism, you could conceivably make that stimulation non-invasively.
Well, here's another problem. So cells of different kinds are all mixed together. So with optogenetics, you could pick out one kind just by making your light-activated molecule go there. Our method cannot easily, as far as we know,
distinguish
between different cells within a cluster. So I think, much as how in the collaboration with Leeway and Emory, the teams had to go from optogenetic drive of a certain cell class to here's what you might watch through your eyeballs. The leap from the discovery to the deployable design might require some creativity and some serendipity.
I'd like to talk a little bit about your overarching philosophy about how you approach science and why you do it that way. One thing you've talked about a few times in public is why the actual moonshot worked in 1962 and why sometimes you're averse to the notion of moonshots and you think that either the term or the effort is misapplied in science.
I like to study the history of science because I think you can learn a lot about the future of science. And I guess one way to think about it is, when is a science ready? If there are thousands and thousands of genes and thousands of cell types, and you want to solve a disease, how do you know for sure which gene or which cell type is the most important? Every now and then you get lucky, but much of the time we're not lucky, right? So you could argue that no brain disease has been fully cured, and the treatments are pretty partial and have a lot of side effects. So that's a clue. It's a clue that rather than doing moonshots where we try to address unknowns without getting down to the ground truth first, maybe we should try to get down to the ground truth. So what do I mean by ground truth? Well, think about the moon landing, right? The original moonshot. When JFK was trying to announce this idea to go to the moon, he wasn't saying, here's this big unknown where we have no clue how to do it. He actually outlined the opposite. He said, look, we've sent stuff into space. Look, if we don't do it, maybe somebody else will do it. In other words, he was trying to persuade people that the moonshot was a good idea because it was feasible. It was built on solid science, in this case, the science of physics. Now imagine doing a moonshot 500 years ago, before calculus, before aerodynamics, before rocketry, before the knowledge of gravity. All the money on Earth might not get you to the moon, right? People might tie kites to chairs or hot air balloons to their chariots and ended up crashing and burning. So what's the right way to do a moonshot? On the one extreme, you have extreme translational science, where the goal is, let's just try to get something into humans quickly. And the other extreme is science that might be very curiosity-driven, but does it connect with an application? Sometimes, but not always. And so what I propose is a third kind of science where we think backwards from the goal, but the goal is not just to do a moonshot without the knowledge of the underlying mechanisms. Let's accelerate the basic science of those underlying mechanisms so we understand what the heck we're doing. So it's kind of fusing the two old models in a way, if you will.
So accelerating the understanding of the underlying mechanisms, nobody's going to want to wait 400 years to solve Alzheimer's.
But look at the examples we talked about earlier. If you build new technologies that let you accelerate your discovery of new things,
precisely,
or make it more systematic, make it more high throughput, make it more comprehensive, then you might be able to make a moonshot without cutting corners. So what if you wanted to make a moonshot to the actual moon 500 years ago? Well, you would probably want to launch a full-scale attack in understanding mathematics, and you'd want to understand gravity, and you want to understand aerodynamics, and then you'd want to get to the moon. So you might actually be able to break it down into an orderly series of steps. Right?
It wouldn't necessarily have taken the full 400 years.
Yeah, you want to start by funding Galileo and Newton and everybody to get, gravity and calculus down quickly. And then you want to get people starting to think about other sciences. One way to think of it is, can we do on purpose for biology what for other fields happened accidentally? Because we know we want to do, but short-term translational shots on goal have been pretty failure prone. I think in brain therapeutics as a whole, the failure rate of drugs to go out of the lab and into the market is well over 90% failure. And in Alzheimer's, if you are talking about drugs that stop the progression of disease, it's 100% failure. But neither do we want to wait 500 years and collect facts one by one. So can we think backwards from the goal, build technologies to let us systematically and comprehensively get to the ground truth underlying mechanistic description of it, and then nail it?
And the race to translation is oftentimes premised on simplifying assumptions, which I know you consider to be very dangerous. You have a great term, the illusion of reductionism. What's that?
Pretending a problem is simple doesn't make it simple. So if I decide to spend my whole life and study one specific molecule that is correlated with the disease, that doesn't guarantee that it will actually be implicated causally in the disease, and it doesn't guarantee that it'll be the most important molecule implicated with the disease. And now from lots of genome sequencing and lots of mechanistic probings of brain diseases, we know that there's a lot of stuff involved. Just, I think, a week or two ago, there was this paper on Alzheimer's disease asking the question of whether gingivitis, gum disease, might be involved.
Really?
And I guarantee that was not on most people's radar screen. So I think the big questions are very often arising by considering hypotheses that are alternatives to the classical ones. And so it raises the question, why can't we just, through new technologies for mapping and controlling in a systematic way what's going on, why can't we confront entire sets of hypotheses so that we can broaden our horizons and not just focus on a single thing?
And you've got something you call the tile tree method.
So the basic idea is, can you have every possible idea of how to solve a problem? I mentioned how we started optogenetics, right? We started by just going through all the laws of physics, and there's only a handful of them, and trying to decide which would be the best for controlling neurons. There's magnetic fields, there's mechanical force, there's light. There's a pretty short list of things that you can deliver into a brain that would convey energy. And then from that, you then consider what kind of effect should the energy have? Will it be converted to electrical signals or chemical signals? If it gets converted to an electrical signal, do you make the molecule that converts light into electricity, or do you find it? So if you think about the logical path that I've been explaining over the last several sentences, we're basically breaking down the space of possibilities like a tree diagram, right? The laws of physics, do you make or do you find, and so on and so forth. And very often, that's how projects will begin in our group. If you're trying to pick a discipline to approach a problem, do you take a chemical approach, an electrical approach, or a computer science approach? And very often, by systematically trying to break down the space of possible ideas into an orderly, structured, pipelined approach that can be tested, it doesn't guarantee that you're going to have every possible idea, but it might reduce the risk of missing something really important, or it might reduce the risk of getting stuck in a traditional way of thinking.
And this is sometimes called morphological analysis, correct?
This is not a new idea, although maybe it's new to apply this systematically in the biological sciences, I'm not sure. But Fritz Zwicky was an astrophysicist who came up with lots of ideas nearly a century ago, like gravitational lenses and dark matter and all sorts of stuff, just by thinking about it in this way.
Really?
And now, of course, people are testing these theories, and these are some of the hottest topics in astrophysics nowadays. But Fritz claims to have thought up these ideas through this tiling tree or morphological analysis kind of approach.
I didn't realize that he thought of dark matter that far back. That's incredible.
I think he did, yeah.
Yeah.
I'm not an expert in astrophysics.
Yeah.
When I got interested in the tiling tree model, I think I stumbled across at least one reference claiming that that was the case.
And all of this, again, seems to point to something that you said at the very beginning, which is the significance of making tools, perhaps before anything else. I think in one interview, you said something like, first get the tools, then get the data, then you can make the theory.
But the tools that we design, it's very important to point out, are designed to get to the ground truth. We're not designing the tools just for novelty's sake. And we want to design tools that will allow us to understand a complex system in terms of its building blocks and the interactions between them. And
tools were not cool, as you said earlier, when you got started. What's bringing them into fashion now?
Well, the tools worked. So I think 2009-ish was when optogenetics started to really scalably take off. And then 2012 was when the U.S. Brain Initiative was initiated by Barack Obama, which said, how can we make more tools?
Was there a connection between those two things?
I think so, because after optogenetics, a lot of people started making tools, calcium imaging, new kinds of microscopes. And then the question became, how can you make more? So optogenetics, I think, might have been one of the first tools that really spread very rapidly in neuroscience. And that's partly because of other driving forces as well. So lots of people were making transgenic mice, or viral core facilities were being launched by universities. So Adgene, a nonprofit, was distributing DNA. So a lot of societal and institutional factors also played a role in helping optogenetics spread very, very quickly. So I haven't done a detailed study of how it spread compared to other neurotechnologies like patch clamp or 2-photon and so forth. But certainly some of the driving forces at universities and the infrastructure of biology helped optogenetics spread very quickly.
Then I know it was a Method of the Year in Nature in 2010 or something like that. And it's intriguing to me that you co-invented that while you were at Stanford doing your graduate work, correct?
Well, it was actually a parallel project that I did in parallel to my graduate work. My graduate work was with Richard Chen and Jennifer Raymond on motor learning. And then Carl Deisseroth and I had met when we were both students and just started brainstorming in parallel. So none of that was in my PhD thesis. That was
really
in parallel to my PhD.
Oh, I didn't realize that. So having done this in your spare time while doing your PhD and with the immediate influence that it had, it is fascinating, as you pointed out at the beginning of the interview, that you struggled to find a post and you ended up at MIT, technically in the architecture department. Is that right?
Well, the MIT Media Lab is in the MIT School of Architecture.
Yeah, that's just remarkable given that this had come out, but that is no longer the environment. I mean, it sounds like there's all kinds of tools development going on now.
Oh, yeah. Then at MIT, along with Alan Jasanoff, I co-founded MIT's Center for Neurobiological Engineering. So we have a couple dozen faculty now devoted to making tools. No, it's become a whole field now.
What are the most exciting things in the pipeline right now, whether in your own lab or in adjacent labs on the tool development front that you think can really help solve the brain?
Well, my dream is to, initially with a small brain, but if it works, then we'll scale it up to larger and larger brains. But starting with a small brain, like a fish or a worm or something, can we image all the neural activity during an interesting behavior, like a decision?
All of it, 100%.
Yeah. And then can we perturb the activity using optogenetic tools in order to test out what activity is important for the behavior? And then can we use our expansion method at the end of the day to map out the wiring and the molecules along those wires? And then can we combine those three data sets into a biophysically accurate, human understandable model of what the brain is doing? So the first building block, watching the brain in action, is a very active area of research now in our group and with our collaborators.
Watching it on a neural level.
That's right. Can individual neurons, for example, blink at you when they're active? Sort of the opposite of optogenetics, if you will. And so last year, we published our first paper in the area of developing fluorescent voltage indicators. In other words, molecules that will light up a cell and allow it to blink at you when the neuron is active. And so the idea now is, what if all these cells in a brain are blinking at you, emitting, let's say, red light? And then with blue light, we go in and activate certain cells and figure out, yep, that activity pattern you saw, that's actually important for something. And then at the end of the day, look at the wiring. Hey, that cell we activated, it drives all these muscle cells over there. That's why the brain controlled the movement. So that's how we connect the dots from structure and function, right?
So this is kind of a dumb, basic question, but you've just given me the answer. I mean, I hadn't realized that there's no way of knowing when a neuron is firing. I guess it makes sense if you can't image them down on the nanometer scale.
And you don't need the nanometer scale, and people have imaged them for a long time, actually. So people have been doing calcium imaging for many years.
And can detect when a spike goes off.
Approximately, yeah. The voltage, of course, is what we really want to see. The calcium is an indirect measure of voltage.
So this
and you don't need nanoscale either. So cells are like 10 microns in diameter. So we think that you don't need to break the diffraction limit to see what cell bodies are doing.
So then if you're watching it using this new methodology, how big of a brain would you start with, like C. elegans or something like that?
Yes, we have several people in the group working on C. elegans, which has 302 neurons.
And for those who don't know, C. elegans it's a worm that is basically a fundamental unit of study because it's been so widely studied. Is that fair to say?
Oh, yeah. Lots of very foundational discoveries are made using the worm.
And it's 312 neurons?
302.
It's 302. So we know where they all are already? I mean, I assume we have a geographic map of that?
An approximate map.
An approximate map of that. And so would that be the nervous system that you might start with?
That, but we're also collaborating with people on flies and fish as well. In fact, we just published a paper a week or two ago with Eric Betzig.
Was that the one on the cover of Science?
That's right, yeah.
Okay, wow. Congratulations on that.
The fruit fly, which has 100,000 neurons, the same as a larval zebrafish. And then we're starting now to think about mice, which have 100 million neurons, 1,000 times bigger than the fly or the fish. And then a human has 100 billion neurons, or 1,000 times the mouse.
So that's a long walk, but you think you'd start with the fly or C. elegans or kind of parallel track the two of them?
And the fish, yeah.
And the fish.
And starting on the mouse as well. So, because if you can solve a small brain, you might learn a lot about what to do before solving an entire big brain, right?
Oh, I'd imagine
you look at the human genome, they solved smaller genomes first and then scaled it up for the human genome.
Yeah, of course.
If you look at cell biology, a lot of the early discoveries were made using yeast, and then they went into human cells, right? Or if you look at a lot of the foundational discoveries of our time, they were initially made using fruit flies or worms because they're small and tractable. And then later they found out that the same principles applied to mice and humans.
So I've only seen the cover of Science. I haven't read the paper, and I hadn't actually realized that was your work. So again, congratulations. Cover of Science is about as big as it gets. What's the thrust of that paper?
Well, the thrust of the paper is that if you use expansion to physically magnify the object, that means you can design microscopes that are kind of clever and that can go at very, very high speeds because the engineering can be focused on volume, speed, and so forth.
Really?
So Eric had invented a technology called lattice light sheet microscopy, and then it's just perfectly matched to expansion microscopy samples.
Wow.
And so that's what we did, is join forces and show that we can map brain circuits about 1,000 times faster than the previous resolution matched technology.
Really?
And so that's just the beginning. Now can we go, what's next, right? Could you go 10, 100, 1,000, 1,000,000 times faster?
Yeah.
That's really why we invented expansion microscopy, because now you can use scalable engineering to image the thing.
Well, I remember there was this really cool citizen science project that was going on somewhere here at MIT that involved mapping out I think, neurons structure of a fly's eye or something like that. So this was a few years ago. EyeWire, yeah.
Yeah, Sebastian Sung.
Okay, so I remember EyeWire, and I remember how much time it was going to take with all the contributions of all these generous people who were contributing their time to map out just part of the neuronal structure of the fly's eye. So now it sounds like you would be able to do the entire neural structure of the entire creature.
Well, we have to do that next. Yeah.
There's
probably more technologies to make it work really well, but that's the goal.
And so then presumably a very detailed map of the neural structure of the fly would be within reach as a result of this work. Then the next step is watching it
But we
light
still
up.
have to label the cells in a way that lets us see all of them. So in this first study, we did not label all the cells. And labeling all the cells is tricky because when they're all labeled, then it looks like a lot of very densely packed things. So we have to prove that.
You have to get to that full labeling. And then having that detailed map, you then get into position to watch when things are firing. Where do you get to the point where you can really discern what's happening within a circuit to really glean, well, that was a thought, or that was a memory that we're able to trigger, or that set of neural firings caused this muscle to twitch?
Well, so it kind of goes back to that tripod technology I mentioned earlier. Image the voltage of every cell, perturb using optogenetics, and then at the end of the day, expand it and get the wiring and the molecules along those wires. I think you need to combine all three data sets, ideally, in order to be able to make a good model.
And the least mature technology of those three right now is which one?
The dynamical imaging.
There's one other thing that I wanted to run through really quickly, because it also seems to be philosophical with you. You sometimes seem to try the opposite. I think that feels like a little bit of a theme in some of your work, certainly with expansion microscopy.
Absolutely. Well, we just published a paper last month on what we call implosion fabrication, where we're doing the opposite to our cells. Have you heard about this?
No.
Oh. So it was also in Science.
All right.
And what we do is, instead of infusing baby diaper polymers in a brain and expanding it to see it better, we take pure baby diaper polymer, no brain, no biology, expand it, and then we photo pattern using a laser different anchors to which we can attach semiconductor particles, metal nanoparticles, and other things, and then we shrink it down. And we can make little wires and electrical circuit-like things.
Wow.
So you can make nanotechnology now with ordinary neuroscience lab equipment. So we call it implosion fabrication.
Implosion fabrication.
Nanotechnology without having to be Intel or whatever. And we probably do the opposite even to ourselves.
That's kind of amazing. And clearly that would be useful to make tiny nanostructure things that you can insert in a brain without perturbing it too much. But that could have enormous ramifications going well beyond neuroscience, right?
Frankly, the stuff we're exploring in-house now have nothing to do with brain or biology. People are always asking, can you make new kinds of circuits? What if people could 3D print computers in their garage? Not just the computer, but the chips too. So I think a lot of the impacts in the short term are outside of biology.
Yeah.
So it's our first non-biology project.
And what is the status of it right now? It seems like you always do a very aggressive job of evangelizing the technology you create and getting it out there so that as many people as possible can innovate as rapidly as possible around that. Have you started that kind of an effort around this one already?
Well, I don't know if we really evangelize so much as teach. A lot of people come to visit us and we'll train people on how to do it. We have a thing we call the Neurotech Training Program where hundreds and hundreds of people have come to visit and learn the skills, and they take it back home with them to their university or their research institution, and they can then use the technology in their science. Yeah, so teaching is a big part of what we do.
Well, again, thank you very kindly for taking so much time this afternoon.
Great.
Congratulations on both the cover of Science and the New England Patriots winning yet again.
Yeah. We take full responsibility for that.
Absolutely. Absolutely. And I will see you soon. All right. We'll now spend 2-ish more minutes with James Joaquin of Obvious Ventures, this time talking about molecular manufacturing. Then I'll be back to tell you about a few upcoming guests and something really cool I have this week for the folks who support this show on Patreon. So James, you call one of the areas you invest in modern materials. How do you define that?
We're looking at high-value materials, an adhesive, a food ingredient, a new metallurgic alloy. Today, these materials are made through energy-intensive petrochemical processes. There's a lot of toxic byproducts and a lot of greenhouse gases. We're investing in startups that invent new ways to make those valuable materials, make them higher performance, lower cost, and most importantly, more sustainable. That could be biological manufacturing, or it could be atom by atom material science construction.
One of your go-to examples is waterproof adhesives. What are those?
There are huge markets for this in the building construction industry, in the marine shipbuilding industry. We need high-strength industrial glue that can hold stuff together, even when it's underwater or exposed to rain.
So let's say I want to make a waterproof adhesive and it's 1984. What do I do?
You'd be working with a lot of petroleum-based raw materials, other chemicals, and you would need to use these energy-intensive processes and refinement of those materials, and you'd generate a lot of pollution along the way.
So what's a more modern way to do this?
You could take nature's best waterproof adhesive maker, the barnacle, and use modern computing to understand which parts of its genome are really good at making a natural waterproof adhesive.
So find the sticky genes.
Sure. And you could then splice those into a single-celled bacterium or yeast. The yeast starts eating plant sugar and then spewing out that really valuable, really effective waterproof adhesive. And plant sugar is basically free because that's most of what's inside corn.
You mentioned modern computing as being part of the process. How long would it have taken to invent one of these materials before the recent machine learning revolution?
Potentially hundreds of years to do sequential experiments to figure out how to make that work.
James will be back next episode to explore a completely different dimension of this domain he calls modern materials. And then later in the month, we'll start hearing from his partners at Obvious Ventures about some other cool areas, like flying cars. Speaking of next episode, my guest is going to be Naval Ravikant. Naval is among the most original thinkers I know in the entire field of tech entrepreneurship. He's both a serial entrepreneur and a very prolific angel investor. In fact, a company he founded, called AngelList, is completely restructuring the ecosystem of capital formation for nascent startups. AngelList will not be the topic of our main interview. But for those of you who support this show on Patreon, it is today's topic. Because while we were sitting around with microphones discussing other things, Naval and I had a fascinating half-hour-plus conversation about AngelList, its history, its philosophy, and the remarkably varied things it's doing to transform the landscape of seed-stage entrepreneurship. If you're interested in how new companies get funded in 2019, this interview will be a terrific resource for you. It's joining the many hours of bonus material that I make available to those who support this show at a level of $5 per month or more on Patreon. You can find all of it at patreon.com. That's patreon.com/Rob Reid, and Reid is spelled R-E-I-D. I'm continuing with my Patreon program despite having my first sponsor for a couple of reasons. The first is that I'm obviously developing a rather exotic and handcrafted form of sponsorship, and it may be a while before I find another sponsor who's willing to engage in that model. The other reason is that I really enjoy creating this bonus material for people who want to dive deeper into the themes of the show. It's a forum for exploring things that may not interest my more casual listeners, and I really like the community of now 1,000+ supporters, which is growing up around it. As for my main interview with Naval, next episode, it's going to break new ground for this show on a couple of levels. First, it will be my first double episode. Naval and I had a very extensive conversation, and I really want to share all of it with all of you. But 60 to 90-minute interviews seem to hit the sweet spot of most of my listeners in terms of duration, so we're splitting this sucker in half. That means I'll be posting 3 episodes this month rather than my usual 2, and I do hope you'll find it's worth your time. And I have a feeling there are some more doubleheaders in the near-term pipeline. The other new thing about this episode is that it's really much more of a conversation than an interview, and that I talk roughly as much as Naval does. The subject is something that matters a lot to me, which is existential risk. And no, that's not the danger of sitting next to a Nietzsche-spouting chatterbox on a cross-country flight, although that could be horrible. It's rather a set of dangers, many of them fueled by advanced technologies in the near-term pipeline, which could, in a worst-case scenario, imperil the existence of our entire civilization. Regular listeners may recall that I wrote a series of essays on this subject on Medium.com this past fall. Naval and I started swapping emails about those essays, and it turned out that we view a number of issues through remarkably similar lenses. And as we started to explore the subject area, it became clear that there was a long conversation to be had, and also that this conversation would be well worth recording and sharing with all of you. So that's what we've done. It's not the most uplifting topic, but spoiler alert, it does end on several optimistic notes. That's coming up later this month. Then I have two fantastic guests lined up in April. The first will be Stanford immunologist Kari Nadeau. Kari is among the world's leading experts in the mysterious epidemic of food allergies, which are now afflicting several 100% more people than they affected just a few decades ago. Kari is on a path not only to cracking the mechanisms and causes behind this grim trend, but hopefully to also finding safe and lasting cures for sufferers. Immunology is a fascinating field. Kari herself is fascinating, and I hope you'll join us. Also coming up, Berkeley AI professor Stuart Russell. Stuart quite literally wrote the book on artificial intelligence, and that over 1,000 universities use the textbook he co-authored to teach the foundations of AI programming to computer science students. Stuart and the broader AI community have not created a monster. Yet. But Stuart is deeply concerned about the long-term risks that AI could pose to humanity. Unlike many of the folks who've sounded the alarm on this front, Stuart is a profoundly informed denizen of the innermost sanctum of the global AI community. He is thoughtfully, persuasively, and courageously on the record about his concerns, which he'll present to the world in a magisterial book, which is expected this autumn. He will preview the core arguments from that book in our upcoming conversation. He will also teach us a ton about artificial intelligence and how it works. I hope you'll join us.